The Mississippi River never stopped moving. 2,300 miles of current draining 40% of the continental United States, pushing 500 million tons of freight past 29 locks and dams every year. The water flowed, the infrastructure functioned, and from the surface, nothing looked wrong. But underneath, the river was losing the system that had kept it clean for millennia. Freshwater mussels, dense, living carpets of them, had once paved hundreds of miles of riverbed across the upper Mississippi. 51 species packed so tightly that surveyors described walking across river bottoms that crunched underfoot like gravel. By 1999, at one of the most productive mussel beds in Wisconsin, density had collapsed from 149 animals per square meter to fewer than 2. In some reaches, field teams pulled up sample after sample and found nothing alive at all. The river was functioning as infrastructure and failing as ecosystem. And the problem ran deeper than population decline, because the animal responsible for cleaning this water has one of the most complex reproductive cycles in freshwater ecology, and the river could no longer support a single stage of it. How do you restart a river's filtration system when the filter itself needs a parasite, a host fish, and a 12-kilogram concrete dome just to survive its first year? The answer involved a hatchery in Wisconsin, a pipeline of parasitized fish gills, and the release of more than 500,000 juvenile mussels, each one smaller than a grain of rice, into a river that stretches from Minnesota to the Gulf of Mexico. The water in sections of the upper Mississippi is turning clear again, and the animal doing it has no brain, no eyes, and no ability to move more than a few meters in its entire life. This is not a rescue story. It is a biological infrastructure rebuild, and the engineering starts inside the animal itself. When the system worked, it worked like a city-scale water treatment plant running on autopilot. Each mussel drew water in through one opening, passed it across internal gills lined with microscopic hair-like structures, and pushed it back out cleaned. A single adult processed 8 to 15 gallons a day. Multiply that across millions of individuals packed into beds stretching for miles, and the maths becomes geological. Think about what that means. A healthy 6-mile stretch strips roughly 25 tons of suspended material from the water column every year. The mussels captured algae, bacteria, and fine sediment, digested what they could, and deposited the rest directly onto the riverbed as dense, nutrient-rich waste. Pumping energy from the open water down into the mud, feeding an entire community of bottom-dwelling insects that in turn fed fish, otters, and diving ducks. And because each mussel buried itself partially into the bottom, their shells interlocked like rebar in concrete, preventing the scouring erosion that spring floods would otherwise cause. That was the system. It was dismantled in 3 stages. In 1889, a German immigrant discovered that Mississippi River mussel shells made excellent clothing buttons. Within a decade, flotillas of clam boats were scouring the bottom with hand dredges. 16 million pounds of shell hauled from Wisconsin waters in 1899 alone, stripping beds that had taken centuries to form. These animals can live 100 years. The extraction rate never gave them a chance. But even after the industry collapsed and clamming was banned, populations did not bounce back because the river itself had been re-engineered. The lock and dam system built across the upper Mississippi over the mid-20th century transformed a free-flowing river into a staggered series of still water pools. That fragmented the migratory routes of the specific fish species mussels depend on to reproduce. It also slowed current speeds, burying the clean gravel and sand that mussels need under layers of shifting, oxygen-starved sediment. And before the system could adapt to any of that, the terminal blow arrived from Eastern Europe. Zebra mussels, introduced to the Great Lakes in 1988 through the ballast water of a transatlantic cargo ship, spread down the Mississippi within years. Here's the thing. They don't target native mussels deliberately. They simply need a hard surface to anchor their threads, and in a river bottom made of soft mud, the only hard surface available for miles is often the exposed shell of a native mussel already sitting there. Thousands of zebra mussels pile onto a single native animal, sealing its shell shut, blocking its ability to feed, breathe, or reproduce. The native mussel doesn't die quickly. It dies slowly, entombed inside its own shell by a living crust it cannot shed. In the most heavily invaded stretches, 80% of native mussels were gone within years. The Higgins Eye Pearlymussel, a yellow-brown green rayed animal no longer than your hand, frequently mistaken for a mud-caked rock by anyone walking the bank, sat at the center of this collapse. Once distributed across 850 kilometers of the Mississippi and 9 major tributaries, its range shrank by more than half. By 1999, sampling at formerly abundant sites yielded 0 living specimens, and you could not simply breed replacements. Because the Higgins eye reproduces through a mechanism so elaborate it sounds invented, and the river could no longer support it. The water was too cloudy for fish to find the mussel. The fish routes were broken by dams, and even if larvae somehow reached a host, the juveniles that dropped to the bottom were smothered or colonized before they grew large enough to resist. The mussels needed clear water to reproduce, but the water could not clear without the mussels. The system was locked in a circle that no amount of legislation, habitat protection, or harvest bans could break. In 2000, a coalition of federal biologists, hatchery technicians, and state wildlife agencies formed a team with a single mandate: build the reproductive pipeline by hand. If the river could not support the mussel's life cycle naturally, they would engineer every stage of it inside a controlled environment and put the finished product back. The first step was the mother. Biologists found egg-carrying females in the few remnant populations still holding on. Using a blunt syringe filled with river water, they gently flushed the specialized gill pouches where larvae develop through the winter, washing hundreds of thousands of microscopic organisms out without harming the mother. Each larva measured less than a millimeter, shaped like a translucent clamp. The female was returned to the riverbed to breed again the following year. In the wild, those larvae would need a fish to survive. And the way the mussel finds one is among the most extraordinary tricks in freshwater biology. The female extends a fleshy flap of tissue from between her shell halves and shapes it into a near-perfect replica of a small minnow, complete with eye spots, coloring, and a twitching motion that mimics a wounded fish in the current. She has no brain, she has no eyes, she has no way of knowing what a minnow looks like. And yet, evolution has sculpted this lure with enough precision that predatory bass and walleye strike at it. When they do, the mussel detonates, blasting a cloud of larvae directly into the fish's mouth and across its gills. The hatchery team bypassed the lure entirely. They placed bass and walleye into holding tanks and poured in a concentrated slurry of extracted larvae, up to 4,000 per liter. The fish breathed. The larvae clamped onto gill tissue. Within minutes, thousands of microscopic parasites had attached. What followed was the critical transformation. Each larva, gripping a gill filament, triggered the fish's immune response. The fish's own tissue grew over the parasite, sealing it inside a pocket. Inside that pocket, feeding on the host's blood, the larva underwent a complete metamorphosis, reorganizing into a fully formed juvenile mussel with foot, shell, and feeding structures. The biologists tracked water temperature obsessively. The metamorphosis runs on a biological clock that counts heat. Every degree the water holds above a baseline threshold gets added to a running total, compounding day by day. At roughly 350 to 400 of those accumulated heat units, the transformation completed. The pocket ruptured. The juvenile let go, dropped off the gill, and fell. In a hatchery tank, it fell to a collection tray. In the open river, it would fall to the bottom and almost certainly die. And this is where the engineering had to solve a problem that biology could not. A biologist named Chris Barnhart designed a device called a mussel silo, a concrete dome weighing about 12 kilograms, 30 centimeters across, with a central chamber of PVC pipe capped at both ends with fine mesh. You load juvenile mussels inside, lower the entire structure to the riverbed, and let the current do the rest. As water flows over the curved surface, it accelerates, the same physics that generates lift over an aircraft wing. That acceleration creates a low-pressure zone above the central opening, which draws water upward from beneath the silo, through the mesh chamber, and out the top. See, the river becomes the pump. No electricity, no moving parts, just fluid dynamics and concrete. The device feeds the juveniles a constant stream of oxygenated food-rich water, shields them from predators, prevents sediment from burying them, and keeps zebra mussels off shells too small and too fragile to survive colonization. Put it this way, direct release into the river produced survival rates in the single digits. Inside the silos, survival ran between 81 and 92%. But the system did not work on the first attempt. Early batches died because biologists had misunderstood what the larvae needed to eat in captivity. Protocols were scrapped and rewritten. Some silos deployed in the river clogged with fine sediment during hot summer flows, choking the upwelling and suffocating every juvenile inside. Each failure erased a generation, because this animal grows slowly, matures slowly, and there were no spare years. By the time the pipeline was running reliably, the Genoa National Fish Hatchery in Wisconsin was producing and releasing over 500,000 juveniles per year. 500,000 animals, each under a millimeter across. Each one the product of a wild mother, a syringe, a host fish, a parasitic attachment, an immune response, a metamorphosis, a concrete dome, and a river current shaped into a feeding pump, all to produce an animal that sits in mud and filters water. The project had manufactured a population. Whether the parasitic life cycle could ever complete on its own, in the open river, without syringes, without hatchery fish, without anyone controlling the temperature, remained entirely unknown. And then came the silence. Freshwater mussels can live 50 to 100 years. They grow slowly, millimeters per season. The 500,000 juveniles deployed into the upper Mississippi did not announce themselves. They buried into the sediment, opened their shells, and began filtering. From the surface, from a passing barge, from a bridge overhead, nothing looked different. The water still ran brown. If a transformation was happening, it was invisible. For years, the only evidence the project hadn't failed was the absence of mass die-offs. Biologists checked silos, measured shell growth in fractions of a millimeter. At the Cordova mussel bed, one of the last places where adult Higgins eye still survived, dive teams continued their annual ritual, descending to the riverbed every August, pulling native mussels from the mud one by one, scrubbing the encrusting zebra mussels off each shell by hand, and placing them back. 75 people one Wednesday a year cleaning clams. That was the state of the recovery. But underneath the mud, the deployed juveniles were crossing a size threshold. At a few centimeters, their shells were dense enough to anchor firmly into the sediment. Clustered together, they began functioning as a unit, hundreds of small shells locking the riverbed in place, slowing the movement of loose sediment, creating pockets of stability. Their digestive waste, expelled as dense mucus-bound pellets, sank into the mud and fed a growing population of bottom-dwelling insects. The base of the food web was thickening around them. Then, in 2005, the evidence arrived. At Prairie du Chien, Wisconsin, Pool 10, the site where sampling in 1999 had found 0 living Higgins eye, biologists pulled up bottom samples and found juvenile mussels that had never been tagged. They hadn't come from the hatchery. They hadn't been placed in silos. They had been born in the river, attached to a wild fish's gills by a female's mantle lure, transformed inside a cyst, dropped off and survived, entirely without human help. The pipeline the team had been running by hand for 5 years was completing on its own in open water. At Cordova Pool 14, the confirmation came formally. Federal agencies reclassified the population as reproducing and self-sustaining. The original mandate had been narrow and desperate: prevent the extinction of one species. But what was emerging from the data was something far wider than species preservation. Now, the mussels were not merely removing sediment. They were pulling excess nitrogen from the water and converting it to harmless gas, trapping heavy metals inside their own shells, stripping bacteria that downstream municipalities would otherwise spend millions treating with chlorine and filtration plants. The river had lost millions of these animals over the preceding century, and the cost of that loss was written into every water treatment budget on the upper Mississippi. As the restored beds grew denser, the cloudiness of the water began to drop. Light penetration measurements showed sunlight reaching deeper than it had in years. Where the light hit the bottom, something else returned: wild celery. Underwater meadows spreading across reaches where aquatic plants hadn't grown for decades because the water had been too dark to support them. The meadows attracted diving ducks. They provided spawning cover for native fish. And here, the system crossed into a feedback loop that sits at the center of everything this animal does. The clearer the water gets, the further a bass or walleye can see. The further it can see, the more likely it is to spot a female mussel's twitching mantle lure. The more fish that strike, the more larvae attach to gills. The more larvae transform and drop to the riverbed, the more juveniles survive to become adults. The more adults filter, the clearer the water gets. The mussels, by cleaning the river, were engineering the exact optical conditions their own reproduction required. A system that, once it reaches critical mass, accelerates under its own momentum and makes human intervention progressively less necessary. The question from the beginning was precise. How do you restart a river's filtration system when the filter needs a parasite, a host fish, and a concrete dome just to survive its first year? You build every stage by hand. You extract larvae from a mother's gills without killing her. You infest fish in a laboratory and track the temperature day by day until the metamorphosis completes and the juveniles drop. You house them inside concrete domes shaped to turn river current into a passive pump. You do this 500,000 times, with animals each under a millimeter across, and lower them into the mud of a river 2,300 miles long. And then you wait, because this animal does not move quickly, does not grow quickly, and will not show results for years. The proof that the engineering held came from two reaches of the upper Mississippi. At Prairie du Chien, where the species had been recorded as functionally extinct, untagged juvenile mussels appeared in bottom samples. Animals born in the river, parasitized on wild fish gills, transformed, dropped, survived. At Cordova, the population was formally reclassified as reproducing and self-sustaining. The loop the team had been running by hand was completing on its own in open water. For less than $1 million in state funding, the project activated a living filtration network that would cost tens of millions annually to replicate with mechanical treatment plants. And each unit will run continuously without electricity for 50 to 100 years. 500,000 animals, each one a yellow-brown lump of calcium with no brain, no eyes, and no ability to move more than a few meters in a lifetime that can last a century. The water is turning clear because they are doing now what their ancestors did for millennia before the button boats, the zebra mussels, and the dams. Pulling the river clean, gallon by gallon, one modest, rock-shaped animal at a time. Sometimes the most sophisticated water treatment system is not something you design. It is an animal that has been filtering rivers since before the last ice age, and the only thing it needed was a way to survive its first year. If that kind of engineering interests you, subscribe. There is more of it coming.